Everybody, my name is Renee from the JQuack Venture Institute. My poster here is to present a new computational software called FRMatch, a cell type matching framework for single-cell RNA sequencing data using the Friedman-Ravsky nonparametric test. Global collaborations, including the Human Cell Atlas and the NIH PRINT initiative in the US, are making rapid advances in single-cell or single-nucleus RNA sequencing. Large amount of data has been generated. However, a major challenge limiting the full utilization of these single-cell RNA-seq data is the ability to compare and integrate data across studies. Here, we focus on the application of mapping the identified cell type clusters across single-cell RNA-seq studies. In the flowchart below, standard workflow has been established from data generation, quality control, sequence alignment, transcript quantification, and cell clustering. Our work is then highlighted in the red box, where after clustering, one branch is to identify marker genes for each cell cluster, and the other branch is to compare the identified cell clusters with new data. We're going to use NS-FIRST as the marker gene selection algorithm, and then feeding that into the new data comparison, where we compare our method FRMatch with two other popular methods, SCMap and Seurat. FRMatch, the method, takes two steps. The first step is dimensionality reduction, where it makes use of the supervised feature selection algorithm NS-FIRST to select informative marker genes, and uses the selected genes as the to reduce the feature space. It takes in two datasets, a reference dataset and a new or query dataset. It selects the same set of marker genes for both datasets. The second step is to perform the matching, and FRMatch is a cell type cluster to cell type cluster matching algorithm. The main idea behind is a graphical model building the minimum spanning trees. As illustrated here in the graph, the minimum spanning tree connects all cells from two clusters in the shortest path. The algorithm then destroys the edge connecting two clusters, connecting cells from two different clusters, which are indicated by different colors. And finally, it calculates the Friedman-Ravsky statistic and p-value. We use NS-FIRST as the feature selection algorithm, and it is an effective and informative way to select marker gene. As we can see, its selected marker genes display on and off binary expression patterns, and in combination, they produce a unique transcriptional barcode for each cell type. The NS-FIRST markers can be two kinds, binary markers or combinatorial markers. The binary markers are those only expressed in specific cell types, and the combinatorial markers are that may express in a couple cell types, but the combination of the expression is unique. We also show here cross-validation studies to compare the cell type matching methods FRMatch, SCMap, and Seurat version 3. We show the LEAV-K cluster out cross-validation under circumstances where one or more cell types is missing from the reference dataset. That is the situation where a novel cell type has been discovered. The LEAV-K out cross-validation proves that FRMatch is robust to missing clusters, aka true negative matches. On the right panel here, Figure A shows the cluster called I5 is left out, and only FRMatch identifies the left out cluster as unassigned here. Other methods produces inappropriate matchings like up here and weakly here and up here. Figure B shows that FRMatch has the best accuracy, the best sensitivity, and best specificity in this cross-validation study. And figure C shows as the number of left out cluster increases from 1, 3, 5, and 7, FR match and one variation of SCMap has the most robust precision record curve. However, the variation of the SCMap has weaker performance in other aspects. So we have more validation results and more details in the bioRxiv preprint of the paper and links to be followed. With real data, we use FRmatch to match brain cell types defined in the full thickness of human middle temporal gyrus neocortex, short for MTG region, with cell types defined in a layer 1 dissection of MTG using single nucleus RNA-seq datasets from the cell type database of the ALAN-BRAINMAP, which is publicly available. In this illustration figure here, the human brain region is marked as star, the region of MTG. The neocortex consists of 6 layers according to neuroscience, and where we focus the comparison between the overlapping layer 1 and the full depth of MTG. Intact single nuclei are then isolated and sorted for SmartSeq library preparation and sequencing. Neuronal cell types clusters are identified in the analysis step. So for the layer 1 dataset, 16 cell types are identified, and for the full MTG dataset, 75 cell types were identified. So the right panel is the FRmatch result. It uniquely maps cell types reflecting the overlapping anatomic regions. In panel A, the rows are MTG cell types, and the columns are layer 1 cell types. Attached to the left is the hierarchical taxonomy of the MTG cell types. Shown in red are two-way matches, yellow are one-way match, and the rest are non-matched. The highlight box A corresponds to a specific subclade of similar cell types that all expressing the VIP neuronal marker, and box B indicates a layer-specific matching of the excitatory cluster from layer 1 to upper layer excitatory cell types from the MTG. And box C suggests under partitioning of the layer 1 astrocytes as an inference from the FRmatch result. And panel B here are examples of matched and unmatched minimum spanning trees. Lastly, we show the comparison between FRmatch and other matching methods in real data. FRmatch here produces the most conservative and lamina-specific mapping of cell types. The other methods produce matching results that have more sparsely distributed VIP types, as shown in box A, or not lamina-specific matchings, as shown in box B. And probably among these approaches, the glial cells in box C were mapped somewhat differently. That is probably due to their overall low sampling and distinct phenotype compared to the majority of the GABAergic and glutamatergic neurons. And our software suites are open source and released on GitHub, both NSFirst and FRmatch. And finally, the FRmatch paper is in press for briefings in bioinformatics, and the preprint is on bioRxiv. Thank you very much.